So, we talked about complete dominance. We're going to talk about incomplete dominance. I'm going to go through this rapidly. You should already understand the process of complete dominance. So we learned complete dominance. You have two phenotypes are possible for three different genotypes. So notice I have three genotypes. 1, 2, 3. Big H, big H, big H, little h, little h, little h. So if I say that hairy toes is dominant to non-hairy toes, these are made up by the way. Hairy toes would be dominant big H, big H. Heterozygous would still be hairy, so only have two phenotypes, two physical characteristics. Now we're going to learn something new. Incomplete dominance. So, when I talk about incomplete dominance, one allele, we know it's a section of DNA, a section of genes that codes for a specific trait, basically, is not completely dominant over the other one. This means the heterozygous is somewhere in between the two homozygous. That's important. That's noteworthy. Okay? And if you want to get even simpler than that, incomplete dominance is basically a blending of sort. Some type of blending. So if I have red and white, the heterozygous would be pink, the in-between. They're incomplete. They are meeting in the middle. There's not a completely dominant one or the other. They blend. So it's a blending of a color. A lot of times it's color. Sometimes it's shape. Most of the time it's color because it's easy to do. Right? I'm not going to ask you like stuff that you wouldn't know off the top of your head. Okay? This is not our class. But, so once again, it's the same idea. Okay? Assign letters to represent the genes. Determine the phenotype, genotype. Okay? It's the same exact idea, but the only thing that's kind of different here is that with this one, it's on your step one, what we did last time was we wrote, okay, red is dominant to white. If it was complete dominance, this would be red. This would be white. Well, in this case, that's not the case here. So what they tell you to do on this, make it easier, assume incomplete dominance, so you have homozygous dominant. Homozygous, homo same, dominant, big R, red. Okay? But the homozygous recessive. Little r, white. The heterozygous is going to be P. Now we have three different genotypes. Big R, little r, big R, little r. Three different phenotypes. Red, white, pink. Clear on that? So if I cross a red homozygous dominant with a white homozygous recessive, big R, little r. That's the same. Everything else is the same. There are examples. Okay? There are some examples. I am not going to do all these examples. They're there. Once again, you have round, oval, or long. So the in-between will be the oval there. Any questions on incomplete dominance? It's just a blending. Simple. Okay? No questions? Pierce, you have any questions?
No.
Nope? Okay. Codominance. So codominance when both alleles of the phenotype contribute. When you think of codominance, what word sticks out to you there? Co. What does co mean?
Together.
Together. Co-author a book. Two authors. Codominance. Two things are dominant. So if you're going to remember, the last one was blending. Okay? A blending of effect. There's no dominance here. This one, if you want to be really rudimentary, really basic about it, spots. If I have brown and I have a white cow, the roan would be spots. Roan is a fancy word for spots. Okay? It is, both are dominant. Both are expressed. That's why it's called codominance. Two things are showing. Alright? So basically, it's not a mixture here. Alright? It's not like this would make a tan cow. That'd be incomplete. If there's spots, it's going to be complete. So the same idea as incomplete. Assign letters. Now, and the important thing, if you have big R is red, and then little r is white. Big R, little r is what they're going to be called roan, which is red and white, spots if you will. It doesn't matter which one you assign to where. Does not matter, you'll still get the same answer. Whenever you assign this in step one, always refer back to what you wrote down. Otherwise, you'll screw it up. Right, Pearson?
Yeah.
Okay. Why don't you go stand up? Well, your eyes are closed. I think different. You stand up if you need to, or just wake up. Okay, thank you. All right. So, good, good, good. So remember, the main thing is that the heterozygous on these, on incomplete and codominance, are their own phenotype. They're different. Okay. Here there are spots. There's a both things are showing up. On incomplete, there's a blend. All right. That is the only thing that I really think that is super important that you understand. Everything else is the same. There's all kinds of examples here. Now, practice problems. I forgot to change this year. All right. So let me make a note here. So everybody turn to this page. Right here. This portion. Practice problems. If you already haven't done it, it's okay. Just kind of chill. This is going to... I need to change the wording on this. All right. Hope I didn't pick up on the microphone.
Yeah.
The back of the background is like doing this noise. Okay. All right. Now, so let me tell you this real quick. Sickle cell anemia. This is equivalent to sickle cell anemia. All right. Marries a woman with the sickle cell trait. This is the sickle cell trait. That is not worded easily for you guys to pick up. So, sickle cell anemia is a little less. And when you use S's, I recommend this, Dalton. This is what I recommend, right? Staring off. See, it's okay. All right, big S. I know you were. I saw that. Big S, little s. I would write them different. Because if you write them like this, a lot of times you guys will get sloppy and you'll do this. You meant that to be little, but it came out as big and then you mess it all up. So being neat is critical when it comes to doing well on Punnett squares. Yes? Be very thorough with your writing. I am done, unless you have questions that we want recorded. No? Thank you, Charlie.